So, very good evening, class. Um, so, yesterday there was some issue with the network, and I just showed the PCR demonstration, how the PCR is done, and shared the respective videos with you. I think here. So, I didn't share the presentation, I guess. Yeah, the PCR presentation. So, maybe I can do this again, and then we switch to the DNA isolation. So basically in PCR, it's a technique done in order to identify specific amount of sequence of DNA, which amplifies for further testing. It's in vitro technique. So in 1983, Dr. Carey Mullis, they developed PCR. In 85, first publication of PCR by Cetus Corporation appears in Science. Then 86, Tag polymerase was first identified. Then Perkel Elmer, they introduces the automated thermal cycler. In 1989, science declares Tag polymerase as the molecule of the year. And purpose was to have a lot of double-stranded DNA molecules of the same size and sequence by enzymatic method and cyclic conditions. So that's the main purpose of our PCR. So it has in three steps, that is denaturation, annealing, extension, DAE. D goes for temperature around 92 to 94, then annealing around 50 to 70, then extension around 72. So at the denaturation, double stranded your DNAs are, they are made into single stranded. And then at the annealing, in this temperature, depending upon the melting point temperature of expected duplex, primers are binded to the complementary sequence, that is your forward and reverse primer. Then in extension, that is an elongation, which goes for 0.5 minute to 3 minute. Here your Taq polymerase, they come and get attached um, to the corresponding forward and reverse primer, and with the help of DNTPs they produce a corresponding sequence. So at the product of extension, after the reaction is done, you will have something like this. So day one, after the first copy, you will have a single copy, and it will go exponentially with 2 raised to power n. After the second cycle, there will be 4 copies. After the third cycle, there will be 6 copies, and so on. So by the end of 35th cycle, you will have millions of copies. And the chemical components that we use during the reactions are 0.5 to 2.5 millimolar, buffer pH is 8.3 to 8.8, and DNTPs are 20 to 200 micromolar, and the primers are 0.1 to 0.5 micromolar. And the DNA polymerase are 1 to 2.5 units, and the target DNA we need less than or equivalent to the 1 microgram. So that was the ingredients. Now the protocol. So first we select the DNA sequence we wanted to grow. Then the primers, typically 20 to 30 base pairs in size. These can be readily produced by commercial companies, can be prepared using a DNA synthesizer. Then thermostable DNA polymerase, example Taq polymerase, which is not inactivated by heating to 95 degrees Celsius. Then thermocycler, which can be programmed to carry out the heating and cooling of our sample over the number of cycles. So afterwards, it could be used for many applications for sequencing, genetic engineering, molecular identifications with respective applications. So these are the instruments that we used. On the left hand side is a simple thermocycler that we see in the most of the molecular biology lab. On the right hand side is the most oldest one. On the left bottom, it's the 4-in-1 thermocycler. So 4 thermocyclers are there within the one machine. And the right one is the most developed, the light cycler, which is having a more advanced functions. So that I discussed yesterday. Some of the students were not able to join and I was not able to record the class. So for them, this will be beneficial. And then there is a video about PCR. This has been already shared over the protocol 1 and protocol 2, so you can understand how practically we do the PCR. um, yeah, and today I actually want to dedicate this day as a book day, so I wanted to share some really good books with you over this platform, that is uh, Wilson and Walker. This is one of the best. This is a Bible. If somebody is looking for research or looking for job perspective, uh, and if they say the lab people or your employer that how many techniques you know, so this, if you go through this whole book, so I'm teaching from this book itself only. So I have divided my work like the basic principle first, then cell culture techniques, then microscopy, then molecular biology techniques, then recombinant DNA techniques. Not this one. Then I have brought this protein structure, purification, and characterization. A little bit about mass spectro also. Then electrophoretic techniques, yeah, the spectroscopy. I mean, this I didn't brought it because they are very specific jobs for them, so in India we don't have many big labs for the same. uh, so you can go for principle of clinical biochemistry if somebody is doing MLT, BLT, um, yeah, drug discovery and development. So this book is very nice. All techniques has been shared in this book with very nice diagrams. This we used to study during our college days actually, for just reading them, making notes out of it. So I will share this book with you. And beyond that, I have some really good books from the biochemistry perspective. Your Lehinger, Lippincott, and Harper. So I have with me, so you can also use them for the later. I have no harm to. It will be good for all of you then, just sharing it. So you have 4 books now: Harper's, Lippincott, Lehinger, and yeah, Wilson and Walker. So I need to share this into the Ames group also. Yeah. Hoot. All settled. So that was about PCR and some book discussion here. So now I will touch the whole actually tools of microbiology from my previous lecture that we have discussed so far, the PCR, electrophoresis, and then there are some macromolecule blotting techniques. That is, you have a genomic DNA. With the help of stiction endonuclease, you can have cut them into small parts, then run over the agrosgel electrophoresis under the negative and positive electrode, and according to the molecular weight, everything your DNA molecule will separate, and then you blot them and transfer with the help of capillary technique over the nitrocellulose membrane. And from there, then you probe the specific phosphorate radioactive phosphor, and then you expose to the X-ray film the DNA that you are looking for. Then you will see the results. So this is divided into 4 parts. That is your snow. Remember this: snow, drop, so S for southern, N for northern, W for western. So D for DNA, R for RNA, P for protein. So in this way you can remember this forever. So southern blotting is done for DNA, northern blotting is done for RNA, and western blotting is done for the protein. So you can omit this, but you can remember snowdrop, write it down, and then S for DNA, N for northern, and western for protein. Cool. So southern blotting is a method for proving the presence of specific DNA sequence. So all these 3 methods, same principle is there. You restrict your specific DNA, then you blot them over the gel, and then you transfer over the nitrocellulose membrane, and then you check it with the help of specific DNA. Northern is also the same. Western is for the protein. It's based on the primary and secondary antibody. So there will be one dedicated lectures on all these techniques. So I'm just giving you like a pinch of salt of these things that what we'll be discussing. Molecular markers, we will also dedicate one day about molecular markers like your RFLP, AFLP. They are very one of the complex subjects. We always tend to keep this at the end of the syllabus because they tend to be a bit difficult to understand, but I will try my best to explain them in as simple as possible. So these molecular markers, they are the one which can help in order to find multiple traits within the from the genome, and it has many advantages over the all other techniques so far. So genetic markers could be short DNA sequence, such as a sequence surrounding a single base pair change, SNPs, a long one, or like mini satellites. So genetic markers includes RFLP, FLP, RAPD, VNTRs, microsatellites, SSRs, SNPs, STRs, SFP, RAD markers, and so on. So the 5 conditions that a suitable molecular marker should include: it should be polymorphic, codominant inheritance, easy and cheap to detect, reproducible, and random and frequently distributed throughout the genome. And molecular markers can be used for several different applications like study of genome, organization of phylogenetic analysis, genetic diagnostic, germplasm characterizations, and so on. This RFP, it involves actually, as name suggests, restriction fragment. You restrict a specific segment of DNA with the help of restriction enzyme, and then you check the polymorphism over the length of DNA in order to find the genetic analysis. In the AFLP, you amplify one specific region with the help of PCR, and then you check the closely related genes in that. In RAPD, it's a random amplification of polymorphic DNA. The same thing will happen, is just we randomly amplify in this particular DNA. We use PCR in it also. Then microsatellite polymorphism, SSRs, simple sequence repeats. So they are the microsatellites which are repeating sequence of around 1 to 6 base pairs, and they are quite highly variable, fast evolving, and quite dominant, whereas they are relatively very expensive and time consuming to develop. And there is a short nucleotide polymorphism, SNPs. So like it shows here that there is CG, and then it's got transformed from TA to CG. So there is both nucleotides from the purines, they made it to the pyrimidines. Purines to the pyrimidines. So could be used for many applications. Then there are short-term tandem repeats in DNA where they occurs in the pattern of 2 or more nucleotides, and they are, just a second. They are the repeated sequences that are directly adjacent to each other, and the pattern can range in length from 2 to 16 base pairs, and used in forensic cases, used for genetic fingerprinting of the individuals. Now comes the main part. So all the techniques that I have discussed with you here, all the molecular markers, right, all the western and northern blotting, so we will do them on the day 4th and day 7th respectively. Yeah, so don't worry, don't think I have shortly explained all these things. We will have a day, we will discuss about them in detail, but today topic I want to more emphasized here is with principle of DNA isolation and purification. So how DNA is isolated and purified further. So DNA could be isolated from any nucleated cell, or any giant anion in the solution. So it's very possible. So basically what we have to do is we have to isolate our DNA based on its characterization. That is, it is not dissolved in the alcohol. Yeah, so based on that, we separate it in our solution. We got a precipitate of our DNA out of the cell. So first we remove the cell membrane to get the DNA out from the nucleus, and then we remove all these pellets, and we remove all the proteins and RNA out of it, and then get the pure protein, and then get precipitated in the presence of alcohol. That's the main principle behind this isolation of DNA. So it has the sources that you can use to get the DNA could be your blood, buccal cells, cultured cells, bacteria, biopsies, forensic samples, body fluids, hair follicles, bones, and teeth roots. So all these things are possible ranges of the same. So, Bachchet's main topic is we are talking about the tools of molecular biology so far. Tools of molecular biology. So far, we have discussed the PCR from the last class, some gel electrophoresis, macromolecule blotting, and then we have discussed today, yeah, molecular markers. Now we are going on over the DNA isolation. That's the main topic. Cool. Students, please do. Yeah, those who have not subscribed to the channel, please do so. It will really appreciate to grow this channel. It's already since it started last 1 year, it has been now like officially be part of YouTube family. So it will really help us to grow this channel and to bring more things for all of you. So please do subscribe, those who have not subscribed yet, and hit like button before, yeah, hit the like button. It will be really helpful for all of us, yeah, to grow this channel. Thank you very much. So these are the various sources of the DNA we have discussed. Now in the DNA isolation, in the routine procedure, these are the three steps that we do. First is cell disruption, then we isolate the DNA, and then we precipitate the DNA. That we have just discussed in the today's discussion before starting this topic. So this is commonly achieved by grinding or sonicating the sample, or removing membrane lipids by adding the detergents to it, right? So isolation of DNA, removing proteins or by adding protease, optional but always done. Yeah, then precipitating the DNA. Then in the third case, we precipitate this DNA. Usually ice cold ethanol or isopropane is used. So since DNA is insoluble, so in these alcohols, it will aggregate together, giving a pellet upon centrifugation, and this will remove the alcohol soluble salt. Then in the cell disruption, this is commonly achieved by grinding or sonicating the sample. So we have discussed. So here we have the sample in our cell lysis. Yeah. Then there's a protein degradation. Then they get binded in it. Then they are washed after the centrifugation. And then after the washing, elution is done, right? So first you put your cell lysed and then go for the protein degradation with the help of proteinase K enzyme. Protein will be degraded and then you wash it with the wash buffer and then you elute it again with the elution buffer and then at the end you get the pure genomic DNA. So the basic rules for lysing the isolation of DNAs, you first lyse the red blood cells with gentle detergent such as Triton X100. Yeah. Then you wash cells, hemoglobin inhibits restriction enzymes, and Taq polymerase. So blood, which first we isolate it, and then we do a gentle like give it a pinch of detergent to it, such as Triton X100, which is most commonly chemical used in the lab. Then you wash your cells. Um, like hemoglobin inhibits restriction enzymes and Taq polymerase, and then afterwards you put them over the ice so that the enzymatic process could be slowed down. Always wear gloves so that the all the our hair follicles or any dust particles should not be part of uh, our protocol. Then autoclave all the solutions and the bottles that you want to use, all your Eppendorf tubes, your standbys, everything need to be clear, crystal, and keep your pallets and supernatants and have the DNA that you're looking for. Then getting to the DNA. So cells that are lyse over the cells in the first you lyse the cells in the presence of sodium chloride so that this sodium chloride helps to stabilize the DNA and remains as a double helix. And EDTA which chelates magnesium ions and is a cofactor for DNAs which chews up the DNA rapidly. Then anionic detergent SDS are added which disrupt the lipid layers, helps to dissolve the membrane and binds to the charge of the chromosomal proteins to release the DNA into the solution. And then we add protease, that is your proteinase K enzyme to digest the proteins in the solutions at elevated temperature of about 56 degrees Celsius to inhibit the degradation by DNases for the 4 to 24 hours. So how to get rid of the proteins? As we suggested, you have your organic solvent extraction and you use phenol into chloroform ratio, like phenol in 24 ratio and chloroform in 1 ratio. Then protein at the interference after centrifugation will be find out at 10,000 rpm for 10 degrees Celsius for 10 minutes. So you will see protein to be having one interference in that once you add these kind of solution to it. So you can get rid of all the proteins out of it. Then you precipitate the DNA by adding 2.5 to 3 volume of ice cold 95% ethanol to the DNA and leave it at minus 20 degrees Celsius overnight. Then you again centrifuge with these specific terms. Then you wash your DNA palette, remove the salt, like your 70% ethanol and air dry. Then resuspend in the distilled water, pH 74, pH 7.4, and then you froze this for like at 4 degrees Celsius. On the long term, you can put at the minus 20 degrees Celsius. So you are good to go with this DNA for further work then. Then quantifying the DNA. How you can quantify? So there is a very nice formula where you have your DNA concentration, microgram per ml, then optical density at 260, whatever you get when you put your sample, then you multiply by the dilution factor and the volume that you have, that 50 microgram per ml divided by 1000. So if you get the value to be coming out to be 50 microgram per ml, so that is your double stranded DNA. If it is 33, then it is a single stranded DNA. If it is 40, then that's your RNA. So based on that, you can differentiate between your different molecules. Then how you can check the DNA purity, that how much DNA that you have identified. The purity of DNA is reflected in the optical density. So you check the ratio of 260 to 280. We check the value of your sample at two different spectrometry units, and then if the ratio is coming out between 1.6 to 2.0, that means your DNA is pure. But if it is coming less than 1.6, then your protein is contaminated into that. If it is more than 2.0, then chloroform or phenols are contaminated into that. Then you have to re-purify your sample. Then in summary, sample for DNA extraction and the lysis of cells at elevated temperature. Yeah. Removal of cellular proteins and precipitation of nucleic acid with ethanol. Then you quantitative and purity measurement of the DNA. So in this way, we can get our isolated DNA. So that's the summary of this. So in future aspect, you can use it for the agriculture development, environment protection, many things could be used for. So we are done with the basics of on today's Friday's lectures, um, with the basics of molecular biology now. I will now show, I mean, I had these virtual labs before, but I don't know if they are working in your case or not. So, so we used to have these virtual labs before, but somehow they are not working. If they are working in your, maybe I have MacBook, maybe that's the reason they are not working. It's saying some problem with Adobe Photoshop. Yeah, always shows this to me. But I will share this link with all of you. If you got time, just go through this link, and if you can play around with that, it will be really great. Good. So we can have the virtual lab another way then. Yeah, Jackson Laboratory.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotech collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotek's proprietary purifying solution, PrepIt, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case, 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case, 14.5 thousand RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples.
Okay.
Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample. And mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Cap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples.
Okay.
There are many components of the
So how does NaCl stabilize DNA double-stranded structure? Mhm. So NaCl actually it's a like it has positive and negative electrode, both ions in it. It's a quite stabilizing agent, and our DNA is a negative strands. Yeah, both strands are negative because of the phosphate group. So the more you add NaCl, the more it will give the stability because of being a negative charge on the DNA. I think that might be the reason behind the NaCl being an active stabilizing agent. Beyond that, I think it's also might help to remove the proteins that are bound to the DNA. So it will attract those precipitants of your proteins aqueous layer and doesn't let attach to the DNA, so that your protein does not get what do you say precipitate along the DNA. Your protein is not the part of DNA. Then you add ethanol or isopropyl alcohol, causing DNA to precipitate. Uh, why different concentration of ethanol use 100 and 70%? because we make everything whenever we are doing any reactions with ethanol, we make it happen in dilutions. We don't abruptly start with, first thing that and that one. In that way, uh, the the errors are very less, and we get really pure part of our DNA. So it's always good to do twice in different concentrations of ethanol. Yeah. So that's it for today, students. We have to do this, the remaining part of our tools in the basics of molecular biology. Um, and we will start from Monday onwards now, or yeah, from Monday onwards, we will start with cell culture and on microscopy. So we will do these two parts. Now we are coming core to the techniques part. So we are done with all basics today, finally. And yeah, now you can make assignments and send it over the email ID. So all the basics that we have done, I have shared you all the notes. To check your notes, you can go to the info, and then there is a files. So all presentations, PPTs are there with you, right? And yeah, some links are also there that we have shared every day. You can also look for them. Yeah, okay then. Thank you very much, everyone. It was pleasure having class with all of you. We see us then on Monday. Yeah, Monday evening, 8 p.m. So enjoy your weekend. Take care. Yeah, bye bye.